So this is the Golgi Apparatus by Kate, Solo, and Mariette.
The Golgi apparatus is situated between the rough endoplasmic reticulum and the plasma membrane in the cell. Its general function is to take in proteins in vesicles, and it sorts them, sometimes modifies them using enzymes, and then eventually secretes these proteins to be sent to the plasma membrane.
So as an overview of the Golgi Apparatus, there are two sides to it. There's the cis phase, which absorbs new material, and the trans phase that secretes material. Proteins from the endoplasmic reticulum enter the cis phase, which can be considered the entry phase, which is usually oriented towards the nucleus. They are transported through the Golgi body and exit from the trans phase, which can also be considered the exit phase, and it faces the plasma membrane of the cell. So this is the incoming transport vesicle. Transport vesicles entering the cis phase of the Golgi apparatus carry proteins and lipids to be modified and sorted by the Golgi apparatus. These transport vesicles come from the rough endoplasmic reticulum.
The cisternae make up the main body of the Golgi apparatus. It's primarily what it's made of. There's sort of these folds and pockets throughout the whole body. They are the location where the modification, identification, and sorting of proteins that enter the Golgi apparatus happens. That's all where it happens. It's not necessarily what does it, but it's the location where it happens. Something else to mention is that the rough endoplasmic reticulum also has these cisternae, and they serve the same purpose. One way that proteins are modified is by the process of glycosylation, in which glycans are created and then attached to proteins and lipids to eventually be sent out to transphase.
Lumen is the volume enclosed by the membranes of any cisternae or a subcompartment of the Golgi apparatus, including the cis and trans Golgi networks. Proteins are targeted for transport to the Golgi apparatus and are transferred from ribosomes on rough ER into the rough ER lumen, which serves as the site of protein folding, modification, and assembly. The lumen can modify the carbohydrate portion of glycoproteins by adding or subtracting individual sugar monomers.
So this is the newly forming vesicle, and once proteins are sorted and packaged for delivery, they begin to split off the transphase of the Golgi apparatus and transport vesicles. It could be considered a gateway for entry of extracellular substances into the cell or for recycling membrane components.
The secretory vesicle is located on the trans surface of the Golgi apparatus, and it forms from the trans Golgi network, and they release their contents to the cell exterior by exocytosis in response to extracellular signals. The secreted product can be either a small molecule or a reported protein, such as a hormone or digestive enzyme. The proteins that the secretory vesicle contains are sent to the plasma membrane.
So this sort of was interspersed throughout the whole presentation, but as far as the Golgi apparatus' relation to other parts of the cell, the vesicles that enter it come from the rough endoplasmic reticulum, and then the vesicles that exit it go to the plasma membrane. So it is directly related to those. In addition, the cisternae, which the Golgi apparatus has, the rough endoplasmic reticulum also has those. So it's directly related in structure to that rough endoplasmic reticulum. As far as how its structure and function are related, it's pretty self-explanatory, but the structure of it is such that there are layers that the proteins move through, and they're modified and sorted through those layers. And if it weren't in those layers, that would be a lot more difficult to happen. In addition, there's vesicles that come in and out. Again, small packages are easy to transport, so that structure is directly related to its function of transporting proteins. These are two real pictures of the Golgi apparatus under a microscope, and you'll notice a big difference between the two of them. The one on the left has significantly more cisternae than the one on the right. The leftmost one is much more common for organisms. Most organisms have between 4 to 8 cisternae, but a lot of single-celled organisms do have as many as 60, and that's what you're seeing on the right, a single-celled organism.
Here are our sources. Thank you so much.